Good evening, everyone. This is Doug McConnell. First of all, thank you, Kate Himes, for setting us up with an uplifting Beatles song. Hande and I were kind of rocking along with that as you were playing. I wanted to thank everybody for joining us this evening. This is a very special webinar for A-Long Swim. This is a really kind of breakthrough news that we want to talk about. I am the co-founder and executive director of A-Long Swim, which is a nonprofit, of course, that raises money for ALS research using open water and marathon distance swimming. It's been successful. It's never successful enough, but it's been quite rewarding so far. I wanted also to introduce my very good friend, Hande Ozdinler, who is the head of the Ozdinler lab at the Feinberg School of Medicine at Northwestern University. I wanted to tell you a quick story because I just realized it was 11 years ago this month when Wendy Abrams introduced me to Hande. And I remember being just bowled over with her enthusiasm and so on. But one of the things that she talked about in that very first meeting was upper motor neurons and why she was convinced that this was going to be a key that would unlock the mystery of ALS. And I was pulled into her enthusiasm and so on. I remember hearing later that that upper motor neuron, the study of upper motor neurons, was unpopular and maybe even worse. Maybe it was even greeted with some derision in the ALS research world at the time. It's been so interesting now to see over that 11 years how this has evolved and so forth. Just several days ago, there was a very important article that appeared in Nature Gene Therapy magazine that highlighted the fact that Dr. Ozdinler's research as it relates to these upper motor neurons really is breakthrough stuff. And the fact is that it is one of the big discoveries is that the upper motor neurons are not dependent on or disease caused by the lower motor neurons, which are in the spinal column, and also that that leads to different kinds of diagnoses and biomarkers and therapies and so forth. So it really throws the door wide open in terms of the study of upper motor neurons. And I just can't be more pleased for Hande and her team and the fact that we can be on that team with her. So Hande, I know you have a few remarks about this and probably can correct how I misstated some of the findings in the article, but please give us a couple minutes on how important this is.
Thank you so much, Doug, for the introduction. And, congratulations to us for our 11 years and many more years to follow. And right. I remember the day as well. And I think our friendship and our collaborations have exponentially grown. And so many things have actually emerged thanks to that friendship and collaboration. More hopefully will be coming. And as you said, this paper has been important for many ways, in many ways. And one is to show that the motor neurons in the brain, which we call the upper motor neurons, do not degenerate just because spinal motor neurons degenerate. And that's an important thing because, especially in the field of ALS, people used to believe that if we cure the spinal motor neurons, the upper motor neurons will be cured because the upper motor neurons die as a consequence of spinal motor neuron degeneration. And that's why they were never the targets for cell therapies or targets for any drug discovery or targets for anything, because they said brain is complex. There are only very few upper motor neurons. They're going to die anyways. Why bother? So now we show actually that the neurons in the brain, the upper motor neurons that actually initiate movement, their degeneration is independent of spinal motor neuron. So that's of the spinal motor neuron degeneration. That's why when you want to build effective and long-term treatment strategies to ALS, you have to include the brain component, the upper motor neuron component into the equation as well. So the paper is a bit complex technically. If you want, I can take you through the figures and try to explain as basically as possible why this study is important, because it will have numerous implications in the future. Would you like me to show some of the figures?
I think so, and don't dig us too deep, but I think that the figures that I saw were highly compelling.
Okay, so let me see if I can share my screen. All right, good. So as you said, this paper was published in Gene Therapy recently, and then I'm very thankful because I'm very thankful to Dr. Svensson, who actually wrote a review about this paper, which was published today again in Nature Gene Therapy, and his comments are very much appreciated, and he tells us that we're actually getting the upper hand in ALS with our results. And okay, so this figure is extremely complex, but I want to take you through to distinguish the brain degeneration and spinal cord degeneration. We have to use a transgenic approach such that we either eliminate gene expression only in the cortex or only in the spinal cord. So this is like Krelok's system. I'm not going to get into details, but this is basically used to generate models where you can erase, as you can see here, for example, the expression is erased specifically at distinct neuron populations, not everywhere in the brain, but very specific neuron populations. See, it's all empty. It's all empty here. So this is done either in the cortex or in the spinal cord. And you can see, for example, these are the spinal motor neurons, and they're black because they don't express that protein. So you can engineer systems such that you can delete or erase something either in the cortex or in the spinal cord. That's very important to distinguish the cortical degeneration versus spinal cord degeneration. So, preparation of this figure, we need to thank Dr. Barış Genç. It took him more than 3-4 years to make that figure, but we made this system where we can distinguish cortex and spinal cord. Then we use viruses to deliver genes of interest, and we use viruses. So I know corona is a bad thing, viruses are bad, but in the lab, you can actually use viruses for delivering genes of interest specifically to the neuron populations. And here, it's again a complex figure, but I will tell you that there are different serotypes of virus, for example, AAV2, 5, 8, 9, different serotypes. And you can inject the virus along the axon, and it will be taken by the axon and carried all the way to the cell body. So then viruses are used as luggage, okay? And you put the genes of interest in your luggage, and you put the luggage in the cargo, and then the luggage will be carried away to specific locations. And here in our case, we actually carried them specifically to the upper motor neurons. So we use viruses to modulate gene expression specifically in upper motor neurons. So that's very good. And we found the serotype that does the best. It's AAV2.
In the process, let me just interrupt for 1 second, Hande. Sure. In the process of doing this, obviously you are working with mouse models and so forth. And I know there has been some complaints about mouse models as it relates to neurodegenerative diseases, and about how the difference, mice and humans just are in different planets, and it's just not going to be worthwhile. Talk for a minute just about the importance of your mouse models and what you've discovered in that regard.
That's actually a very good question. So usually mouse models are generated to understand the cellular and molecular basis of diseases, because if you look at the mouse and human at a species level, it's very different, right? But at a cellular level, they are very, very similar, almost identical. And at a genetic level, that homology is more than 95, 96%. Especially some genes are 99%, it's almost at the genetic level, the similarities are very remarkable. At a cellular level, similarities are profoundly the same. When it comes to upper motor neurons, the neurons of interest in our lab, the upper motor neuron in a well-characterized mouse model is almost identical to the upper motor neurons in ALS patients or in HSP or PLS patients. So it depends on how you look at it. If you compare mice and human, yes, they will be different. And if you expect that increasing the life expectancy of mouse with a compound or with a drug is going to increase the life expectancy of a patient, it may be different. There may not be translation to species level, but at the cellular level, they are identical. So this is also shown by Dr. Gautam in the lab, and he's also the Ellen Fellow, the Longswim Fellow. He has done remarkable studies, especially with electron microscopy level, taking the upper motor neurons from well-characterized TDP mouse models and also ALS patients with TDP pathology, and they were identical. So that's why we feel more comfortable when we focus our attention to the neurons that degenerate and not just study the mice. So we are studying the upper motor neurons at a cellular level. And in that case, we think that if we find ways to improve the health of upper motor neurons at a cellular level, this may translate to patients in clinic.
Right. Okay. I interrupted your slides, and
no problem.
While you're going to the next one, I just want to reiterate, Kate Himes just put in the chat box. If any of the listeners have questions, and you inevitably will, please just put them right into the Q&A box at the bottom banner of your screen. And as we get toward the end of Hande's remarks, we'll see if we can kind of combine some of the questions and ask her. Anyway, Hande, please go ahead.
Yes. So as we followed, now that we separated cortex from spinal cord, and we have the UCHL1 null model in our lab, in which the upper motor neurons show profound degeneration. And here are the upper motor neurons. You can see they have large cell body. They have a very long apical dendrite. This apical dendrite is very important because it is the site where other neurons talk to the upper motor neurons. So it is a site of connectivity. And in diseased neurons, you can see that this apical dendrite is filled with vacuoles. They don't have spines. They can't talk to each other. And now when we actually deliver genes to these diseased neurons, the gene that they lack, which was UCHL1 in that case, boom, they become better, they improve, okay? And so that's—
So is it safe to say, Hande, that with this treatment, they're actually, the deterioration is reversing?
I don't know if it's reversing, but they lack a gene, and because they lack that gene or they lack that protein, they become diseased. So when you give them the protein that they lack, they say, oh, okay, this is what I need. And then they are actually, regain the integrity of their apical dendrite, the integrity of their cell size and soma. So they are relieved in a sense. So
Wow.
this also shows us the importance of gene therapy or gene delivery, because if we actually can identify what is it that the upper motor neurons need for survival, and if we can develop ways to give them what they need using AAV or viruses for delivery, then we can actually relieve their pain, relieve their suffering and make them happy, right? So that's why the gene therapy offers many advantages. And again, here I can show you the healthy apical dendrite with all the spines. And neurons communicate to each other through this connection. Spines are at the site of connections. And when they are vacuolated, when they're all filled with vacuoles, you see there are no spines left. So if you have an apical dendrite like this, it's impossible for you to communicate. It's impossible for you to be listened or to be heard or to be in the circuitry. So in that case, if the brain cannot speak to upper motor neurons, the upper motor neurons cannot really initiate action potential. They cannot tell spinal motor neurons what to do. So, they can't initiate movement. So
Right.
to initiate movements, we have to turn these vacuolated apical dendrites to the healthy condition. Okay. And then our proposal was to use gene delivery. Our proposal was to say, all right, maybe we'll give you the genes that you need, and we will try to improve your stability. And that's what we did with UCHL1, because that was the UCHL1 null mice. So that was very significant. And all these dots here are in independent mouse. You can see N is like our N numbers are pretty high. And you remember the question that you asked, the mouse and human may be different. And here, for example, I'm showing you the apical dendrites from human patients. So these are the apical dendrites of not mice. These are human. And look, familial ALS cases, FTD ALS cases, sporadic ALS cases, look at their apical dendrites. They are very similar to the well-characterized mouse models. And again, we show that at a cellular level, what we see in the mouse is also observed in the human. That's why improving, especially the apical dendrite integrity and spine health, would be very important for cortical connectivity. And that's what's missing in ALS patients and HSB and PLS patients.
I'm glad you mentioned HSP and PLS as well. This really is applicable to all neurodegenerative disease, is it not?
I think so, because we call upper motor neuron diseases are diseases in which upper motor neurons degenerate, and HSP, PLS are definitely important components of that. And ALS is also one of them, because in ALS, in addition to the upper motor neurons, spinal motor neurons degenerate as well. But in HSP and PLS patients, you know, the cytoarchitectural defects, mitochondrial defects, ER defects, axon transport defects are vastly observed. And, you know, we also observe them in the mouse models at a cellular level. All right, so now I showed that giving UCHL1 to the UCHL1 null mice, which lacked UCHL1, helps them, which is, you know, no brainer. Of course, if you don't have it, we give it, it's going to be good, right? But then we took one step ahead and we said, all right, we know SOD1 is mutations of SOD1 leads to ALS and upper motor neurons degenerate in the SOD1. If we give UCHL1 to the SOD, which is not related, right? Because it's not a UCHL1 mutation. This is a different concept now. And guess what? When we give UCHL1 or when we express UCHL1 gene in upper motor neurons that are diseased due to misfolded SOD1 or SOD1 mutation, they also improved. So that's big deal.
Wow.
Because it's not that we're giving SOD1 to SOD1 mutant. We are giving UCHL1 to the SOD1 mutant, and it improves apical dendrites. Look at the apical dendrites, right? It is improved. Normally it's 60-70% are with vacuoles, now only 20%. So that's pretty good. And then of course some diameter enlarged and so forth. So this was very good. But then we said, okay, if we try a different mouse model, a different disease mechanism, how about TDP pathology? And then you observe TDP pathology in about 90% of ALS FTLD cases, in a vast majority of ALS cases. So this is a very important pathology. It's also observed in HSP and PLS. And the Balo mice is a good representation of the TDP pathology. And this mice shows progressive degeneration of the upper motor neurons. And that's the paper that Mukesh Gautam published in Acta Neuropathologica, that the human and mouse are similar at a cellular level. We said, okay, how if we give UCHL1 to the TDP? Totally different. Because in the patients with SOD1 mutation, there's no TDP pathology. So there is a distinct distinct biology or pathology between SOD and TDP. They are not overlapping so much. So this is totally different. And we said, okay, let's give UCHL1 to the TDP. And guess what? Even though in this mice, it's not the UCHL1 gene that was mutated, you again see improvement in apical dendrite stability. You see improvement in cellular cytoarchitecture. So this is helping the upper motor neurons to stay intact, to stay healthy, to retain their cytoarchitecture, and they don't show the phenotype of the diseased or degenerating neurons. So up to here, this is really nice. And
Hande, on this same issue, irrespective of whether it's SOD1 or a TDP and so forth, one of the things that I pulled from one of the articles was that the early warning of some of these deteriorations really before there are, and again, these are early warnings of the upper motor neurons versus the spinal cord neurons and so forth, that might give us a little bit of a head start on anticipating somebody starting to deteriorate with ALS symptoms. And my follow-on question to that will have to do with biomarkers and how we ultimately will be able to test somebody for that. But talk for a minute about this early warning, advanced warning thing.
Yeah, so for example, our colleagues in Australia, Dr. Wookie, Kiernan, and their groups have shown that there's cortical hyperexcitation, which means the cortical neurons fire like they go wild, and that actually happens even before symptom onset in ALS. So in our view, the cortex actually raises the red flag, saying like there's a problem, much earlier than symptom onset. So if we can detect the cortical hyperexcitation and cortical connectivity problems and so forth, that would be very useful. And even our colleagues in Australia suggest that this cortical hyperexcitation could be an early detection marker for ALS pathology. So, of course, more work needs to be done on this aspect, but I think the cortex really speaks to us, and cortex really says, there's really something wrong and I can't really initiate movement. There's the motor neuron circuitry is not functioning properly. So, of course, our goal is to detect biomarkers to show the timing and extent of upper motor neuron loss in patients. But in some patients, the disease has a genetic component and they already know the mutation. And this is usually true for, let's say, HSP or PLS patients or some ALS patients. And especially, for patients who know their mutation, it is important to determine whether correction of that mutation or giving them the wild type form of the gene that is not mutated, would that have any clinical implications? Would that really make them, make their upper motor neurons healthier and function better and, stop degenerating? Because, for example, KIF5A mutation, and in that mutation, there is a problem with axon transport, right? The axonal degeneration and so forth. So if we know the mutation and we know that this mutated protein cannot function, is it possible for us to give the functioning protein by gene delivery directly to the upper motor neurons by direct cortex injection? Would that be a therapeutic application? So in my opinion, that it would be. We may not be there yet, but I think that's where we are going, especially for patients with known mutations and especially patients with rare diseases. Because if a disease is very rare, it's very hard to have, full-fledged drug discovery with, 400 patients in phase 3, or it's almost impossible if there are only, 50, 60 patients in the whole world with that particular mutation or with that particular disease. How are we going to have regular drug discovery studies for those patients? And especially if a disease is, takes a long time. Let's say you have PLS, but you have PLS for 20, 30 years, right? And it's a slow progress. So then drug discovery companies may not be so much interested, because then they have to do the clinical trial for how long? Like 2 years, 3 years? That's going to be so expensive. And plus they only will help 5 patients. So they go like, okay, that's a tough decision. But we can move towards personalized medicine approaches. And rather than curing the disease, we cure the patient. So we, tailor our treatment strategies around the patient. We identify which mutations they have. We identify what is the cellular pathway or canonical pathway that is affected. Maybe we can actually find a solution for the patient, and that we can actually develop, gene therapy approaches again for the patient. I think we were not there, 5-6 years ago, but we are running towards that goal very fast. Everything is aligning, and I think in the very near future, this is what we will be able to accomplish.
And do you think the complexity of ALS itself is one that really is logically lends itself to this personalized? I mean, this is very exciting stuff for everybody who's on this call, I'm sure, but it's just to contemplate that you could have a custom solution for one person's motor neuron behavior and so forth, and really treat the precise sort of issues that one person has. That's almost incomprehensible to us lay people.
I think so. I think that's where we are going now, with specificity of different serotypes of viruses, and with our better understanding of cell type specificity, selective vulnerability, the underlying causes of vulnerability. It's taking us there. And also the need, because as you said, the ALS the disease is very heterogeneous, and there are numerous underlying causes, and patients may be developing the disease due to different underlying reasons, right? So right. Then, one solution is not going to be a solution for everyone. So it took us a while to realize how complex the disease is, because when we find something, we think that everybody's going to benefit from this, or one drug is going to, cure all patients. When we realize that's not the case, so then, I think we need to understand why patients develop the disease, and that's why we need the biomarkers to, subgroup patients, and then also even for the most rare forms of the disease, maybe we go even more personal approaches. So I think that's where the future is going, and especially with gene therapy to upper motor neurons are very important, I think, and they're also accessible, more accessible than the deep brain stimulation or deep brain targeting that they have done for the Parkinson's disease. Like you have to go all the way to the cortex, to the for Parkinson, you actually have to almost take a cross-sectional, right? You need to go really deep. But for this one, you only need to go to the layer 5 of motor cortex. So it's not very deep. And the surgery that is required is, much easier than deep brain stimulation that they have used in Parkinson's patients. Of course, right. It's not easy, but it is less cumbersome. Let me put it
and less invasive.
Yes, less invasive. Yeah. Yes,
it's interesting. Not to be too maudlin about this, but my father was diagnosed with ALS 30 years ago next month. And I remember he was a practicing veterinarian and was very candid about his diagnosis and what the prognosis was for it. I couldn't believe what he was telling me, and that there was really no hope for this thing. And he sort of shrugged his shoulders and he said, and there never will be, because there's no money in it. And I think now, fast forward 30 years, hearing you talk about personalized medicine and being able to diagnose this right down to the individual's opportunity, what that tells me is maybe there will be some money in it. And that's so the old man, this is how we prove the old man wrong, I think.
Yeah, I really think in the near future, there will be many options for ALS patients, HSP patients, PLS patients, and I think gene therapy will be one of them.
Wow, this is exciting stuff. Kate Himes, I know that you are collecting some of the questions that people are asking, and I've been too focused on Hande's remarks. Do you want to talk a little bit about some of those remarks?
Yes. So we've got a question asking, can neurogenesis regenerate upper motor neurons?
I'm happy that you asked this question. So I was actually a postdoc in Jeff Macklis's lab at Harvard Medical School, and Jeff Macklis was working on neurogenesis of postnatal brain, and he was working on upper motor neurons, corticospinal motor neurons, and he showed that there is neurogenesis, albeit very small levels, very small numbers, but he was able to show one or two. So I don't want to say it doesn't happen. I think it does happen, but it does not happen to a level that you can replenish the upper motor neurons. For example, the neurons in the olfactory bulb, the SVZ region, they continuously are reborn. And also in the dentate gyrus of the hippocampus, there's regeneration. That's not to the same level, but I think there is some, even though very small levels of upper motor neurons reborn. Yeah.
Nice. Can you explain what HSB and PLS are?
Yes, so the HSP is hereditary spastic paraplegia, and PLS is primary lateral sclerosis. So these are diseases of the upper motor neurons. And even though the upper motor neurons degenerate in those diseases, the underlying causes of degeneration could be different. PLS is considered a spectrum of ALS, but HSP has its own genetics, has its own mutation, and they're considered in a sense different diseases. But at the end of it, the upper motor neurons degenerate in those patients.
Hande, is it safe to say that what isn't toxic to mice is also safe for humans?
What is toxic to mice is safe to humans?
What is not toxic to mice?
I don't think that I would say that. When it comes to toxicity studies, it's a broad sentence. I don't want to say that, but I think what is toxic to mice could be toxic to human. And if it is not toxic to mice, I don't think it doesn't mean it's not toxic to human.
Great. So we've got a question. How do you test me to see what gene I need?
Okay, so usually, I don't know if this is performed on a regular basis, but there are sites where you can look for, like, say, mutation analysis, GWAS analysis, to see what mutations you may have. This, I don't know if there are services at this level or if it is just at a research level, but if your mutation is determined by a genetic study, that's how you know what your mutations are. And I wish that there was actually a site where you go give your DNA or blood, and then they tell you, okay, this is, they run your gene list and say, all right, these genes have mutations. I don't know if there are services like this. So we do it at a research level, but I don't know if those services are given to patients.
Would this help with people who have HSP also?
I would like to say so, because for HSP and PLS, again, the upper motor neurons degenerate, and we find that the upper motor neuron degeneration, at a cellular level, is the cytoarchitectural integrity. The apical dendrites are very important for the health and function of upper motor neurons. And in HSP and PLS patients, apical dendrites also degenerate. So if we find a solution which would improve their integrity, I would say that it would have an implication. And also the health of mitochondria, ER, protein aggregation. These are all common problems that are shared by diseased upper motor neurons. So, there could be personal and unique problems, but there may be also common and shared problems. And I think both needs to be investigated to develop a more personal and more effective treatment strategy.
This final question is a long one, so bear with me. So a team at the University of Illinois, led by Thomas Gage, just published a paper in Science Advance. They did some gene silencing on astrocytes in the spinal cord of mice with ALS. They show some improvement in mice. Is it too far from the origin of the disease? Historically, the classic approach with ALS was to target the spinal cord.
Okay, so this is actually a very nice paper. And they try to understand the impact of astrocytes, microglia, the non-neuronal component in the central nervous system. because the motor neurons are actually friends with astrocytes and microglia. And there is this molecule which we call glutamate, and it is important for neurons to start action potential, for neurons to be active. But too much glutamate is not good, because then they're always active, okay? So astrocytes, they take up this glutamate and they say, all right, I'm going to clean it for you. And, I'll take it so that you don't become so active. So there's a relationship between astrocytes and motor neurons. And it's a very friendly relationship. But during disease, astrocytes also are affected and they stop cleaning that glutamate. So when they stop cleaning that glutamate, the neurons become like angry and very active, okay? So for example, one of the drugs that is used in clinic for ALS is riluzole. And riluzole actually dampens the astrocyte activation and, being unhappy. So we have to keep that balance between astrocytes and motor neurons, all right? So that they become friends or they stay friends for a long time. And in that study, they wanted to see what is the impact of the astrocyte on the whole motor neuron circuitry. And then they realized how important they are, right? And I think something similar is happening in the motor cortex. And Dr. Gautam actually showed this with activated astrocytes, microglia, innervating or cells that innervate the brain parenchyma from the bloodstream. So I think that in addition to the neurons, we have to also make the cells that help the neurons partner in that improvement, so that we should also make the astrocytes happy within the motor cortex too.
One last question. Sorry, Doug.
Okay.
Can stem cells replace upper motor neurons?
Yes, so that's a good question. The answer is no. The stem cells, okay, so let's define stem cells. To be honest with you, each one of us, every one of us, were a stem cell once, right? So when the sperm and the egg unite, and you have the first cell, and then the cell division starts, to occur, and those cells have the potential to become any cell. So stem cells are the cells that have the potential to become any cell. And with correct manipulation or with correct modulation, they can be turned into different lineages, different cell types. And stem cells do make upper motor neurons, because we were all stem cells once and we all have upper motor neurons. So stem cells, in theory, some of them, by some unknown mechanisms, must be turning into upper motor neurons. But right now, we don't know how to accomplish this in a dish. So there are many stem cell cultures, and they are either generated from, neural epithelial cells or blood cells, or there are many different sources to generate stem cells. And you generate, you change their gene expression profiles and so forth. And they make spinal motor neurons, they make astrocytes, they make interneurons, glutamatergic neurons. But so far, the efficiency of making upper motor neurons has not been very successful. So that's why in our lab, rather than using differentiated cells, which do not have the properties of upper motor neurons, we actually use the fluorescently labeled upper motor neurons, which are indeed the upper motor neurons, and they are very similar to the upper motor neurons in humans. So, so the answer is, in biological terms, yes, stem cells must be differentiating into upper motor neurons, because we have upper motor neurons and we were a stem cell once. But in the dish, we are not there yet, and we cannot really produce upper motor neurons in good percentages and in good quality. So that's why I think more work needs to be done in that area.
I think that, and as time goes on, it's interesting to see how Of course, there's a very active stem cell track in ALS research. I think as time goes on, my observation has been that these various tracks are really coalescing around half a dozen different areas. Upper motor neurons clearly is one, stem cells is another. And I think that my observation is that those have evolved and developed and coalesced really around collaboration among different centers. I mean, and of course you collaborate within the four walls of Northwestern, but also with the University of Illinois and with Harvard and with centers all over Europe and Australia and so forth. I remember that collaboration really was one of the big sort of points of agreement with us when we first got to know each other. I know we're running a little bit over, Hande, but can you give us just a minute on some of the collaborations that you have and how important those are?
Yes, I think, as the diseases are very complex, like ALS is a very complex disease, and the motor neuron circuitry is also very complex. I don't think that there would be just one lab which reveal the biology, pathology, connectivity, everything just by themselves. So it is required that we work together. And in that sense, our work is also very, it requires collaboration with other labs because, there are things that we know, but there are many things that we don't. And there are things that we can do, but there are many things that we cannot do. And in that sense, yes, we collaborate broadly within Northwestern, within Chicago, within United States, and also globally. And actually, I was just writing a paper and we're going to submit tomorrow. For example, it's a collaboration with our Italian colleagues, and we're just writing a paper about metabolites and how metabolites change in ALS patients with alsin mutation. And now, for that one, we're collaborating with doctors in Italy. And there will be another paper, and we're collaborating with people in Australia. So yes, for the scientists, I think the world is our playground, because the diseases do not care about nationality, right? They don't care about where you were born, what language you speak. And I think to overcome such complex and diverse diseases, we, the scientists, must also work without borders. And that's what we're trying to accomplish.
Well, we are so thrilled to be on your team. And are so proud for the discoveries that you've made, these publications, and the wide sort of response that the scientific community has been having to these discoveries. We just couldn't be more proud for you. I also noticed that we're 45 minutes into a 30-minute call, and so my apologies for running over. I want to thank everybody for joining us and so forth. By the way, A-Long Swim is now dedicated to really having a regular schedule of scientific webinars like this one. We're hoping to have, in fact, we have them planned for both February and March, and we will keep everybody posted on kind of what the subjects are and so forth. We sure hope that you can join us for each of those. And Hande, any parting words, and then we'll let these folks be on their way.
Thank you so much. So I'm actually looking at the questions and answers. There's one question from Henry Chupi, and he says, I don't know of a mutation for PLS. We actually have a paper published about the PLS genes and also the proteins that are encoded by them. For the PLS, for example, Arlin2, Parkin, FIG4, SPG7, DCTN1. So those are the genes. And more genes are, of course, identified, almost every other month. And the more we understand the genetic basis of these diseases, we will have better treatment options. And I think the gene therapy is going forward to be one of the options in the future.
That's great. That's great.
Thank you so much.
Thank you again, everybody. This was wonderful. Thank you for joining us. Watch your email for invitations for February and March, and let's keep this ball rolling because this is really exciting stuff. And again, Hande, on behalf of all swimmers out there, we could not be more proud for you.
Thank you so much.